Okay, this is lecture 5. It's on microbial metabolism. I should point out to you that lecture 4 was an introduction to this lecture. The material covered here in this lecture is partially also an expansion of what you did cover in anatomy and physiology last year. We did talk about cell metabolism in that course, and microbial metabolism is not that much different. It covers the same kind of major topics, and here there's more detail. Now, some students are somewhat put off or stressed, for want of a better word, by some of the detail shown in these slides, but what I'm going to do is focus your attention on the important facts. Don't get lost in all the detail that you see on these pathways. I'm going to focus you on what you need to know and understand to give you a broader overview. There is detail, but not quite as much as in all the slides, and I'll point it out to you whenever that comes up. Okay, first of all, most microbes use carbohydrates as an energy source. For example, glucose. It could be some other carbohydrate, but most of them use carbohydrates like glucose to generate energy. And the main two processes or metabolic pathways that are used to generate that energy are cellular respiration and fermentation. And cellular respiration involves the use of oxygen and includes a process called oxidative phosphorylation. And fermentation does not involve the use of oxygen. So these are two energy producing processes that cells can use. Not all cells can carry out both of these. Some cells can only carry out one or the other, but some types can carry out both. Remember, energy produced will be produced in the form of ATP because that's an organic molecule that these cells can that stores a lot of energy, and cells can easily use as a source of energy by releasing it quickly when they break one of the phosphate bonds in ATP. Now, there are 4 major stages to the process. First, we're going to deal with cellular respiration, not fermentation. First, we'll do cellular respiration. So essentially, we're talking about a process of oxidation of carbohydrates. If you notice here, each step listed here is an oxidation. Okay. In each step, you have production of some sort of product, which then goes on to be oxidized further in the next stages. So there are 4 principal stages in cellular respiration. They are glycolysis, pyruvate oxidation, Krebs cycle, and the electron transport chain. I'm going to go into detail in the next few slides on each of these, but just as a summary to start you off, in the oxidation of glucose, the product of that process is, the end product is pyruvate or pyruvic acid. Along the way, high energy compounds are produced, including ATP and NADH. So these are 2 molecules which contain a lot of energy, ATP and NADH. Of course, ATP, that's what the cell wants, and that's the end point for that. But the NADH will be used later on to generate, the energy in NADH will be used further down to generate ATP. Okay. So we have glycolysis, the oxidation of glucose to pyruvic acid, also known as pyruvate. And then that pyruvate or pyruvic acid undergoes oxidation. So we have first we have glycolysis, and then we producing pyruvic acid, and also in terms of energy, ATP and NADH. And then the pyruvate or pyruvic acid undergoes oxidation in pyruvate oxidation to form acetyl-CoA, and in terms of high energy compounds, more NADH. So here in the first line here, okay, in the first line shown here on each of these, this line, that's what's happening to the carbon containing organic molecule each time they're undergoing oxidation. And the second line, that's what's happening in terms of production of high energy compounds. So the first stage is glycolysis, oxidation of glucose to pyruvic acid or pyruvate, along with, in terms of energy, production of ATP and NADH. The second stage there, the pyruvate or pyruvic acid undergoes oxidation to form acetyl-CoA, along with production of NADH. And in the third phase or third stage here, we have the Krebs cycle, where the acetyl-CoA gets oxidized, and it gets fully oxidized. It keeps going round and round in a cycle, getting converted into different molecules, each step in oxidation. And as that happens, in terms of not the carbon skeleton, okay, remember the first line is, the first line here for each of these is what's happening to the carbon skeleton organic molecule, and the second line is what kind of high energy compounds are being produced. So along the way, during the oxidation of acetyl-CoA in the Krebs cycle, you get production of three things in terms of high energy. One is ATP, another is NADH, and also FADH2. Now, NADH and FADH2, which are, what should I do here? NADH and FADH2 produced during glycolysis, pyruvate oxidation, or the Krebs cycle will all be, they can all undergo oxidation. So each of these, and now this is an energy producing step, okay? So all of these can undergo oxidation and produce energy by oxidation of NADH and FADH2 to produce ATP. And this occurs in the electron transport, just a minute, the electron transport chain. So you have oxidation of NADH, FADH2 for production of ATP. So we have ATP in glycolysis, we have ATP produced in Krebs cycle, and we have a lot of ATP produced during the electron transport chain. Okay? 4 principal stages for cellular respiration: glycolysis, pyruvate oxidation, Krebs cycle, electron transport chain. The glucose is converted by oxidation to pyruvic acid in the first one. Pyruvic acid is converted to acetyl-CoA in the second. Acetyl-CoA becomes fully oxidized as it goes round and round during the Krebs cycle. And any NADH or FADH2 that is produced in these three will be used for production of ATP in the electron transport chain. This is a diagram that goes over everything that I'm going to discuss today. And here we're discussing cellular respiration. Earlier I mentioned, and this occurs in the presence of oxygen. Earlier I also mentioned fermentation is another way that that cells could produce energy, but we're not going to talk about that yet. On the right-hand side in this diagram, everything involved in fermentation is described there in a sort of a flow chart. So in here, that's fermentation. And on the left is cellular respiration. Notice that glycolysis, the oxidation of glucose to pyruvic acid, is common to both of these processes. After that, they go in different directions. This production of ATP is happening really during glycolysis, so it will be in either respiration or fermentation. And this NADH is happening, production of NADH is happening during glycolysis. And so it's happening also for fermentation and respiration, but it gets used up in fermentation. It gets used up, and that arrow shows it being used here for fermentation of the pyruvic acid. So there's no net production of NADH during fermentation. There is NADH and ATP produced in respiration, and there is ATP produced in fermentation. Let's stick to respiration for now. As I told you earlier, the first stage is glycolysis. What color can I choose here? The first stage is glycolysis, which involves the oxidation of glucose to pyruvic acid. Then pyruvic acid is oxidized to form acetyl-CoA. Then acetyl-CoA undergoes oxidation completely as it goes round and round during the Krebs cycle, and I'll explain what round and round means later. Okay. And along the way, during glycolysis, NADH and ATP are produced. In pyruvate oxidation, NADH is produced. In the Krebs cycle, ATP, NADH, and FADH2 are produced. Now, NADH and FADH2 are high energy compounds. They have taken up electrons. They are electron carrier molecules, and they will go here and be used in, see, all the NADH that's produced will go to the electron transport chain, where it undergoes a process of oxidation reduction reactions, redox reactions. And at the same time, there will be chemiosmosis, and I'll explain what that means later. The electrons that are carried in to the electron transport chain are transferred from one molecule to the other. That's why it's called the electron transport chain. They get transported from one molecule to the next molecule to the next one, like oxidation reduction reactions, until they reach the final electron acceptor, which is oxygen. That's why cellular respiration only occurs in the presence of oxygen. And the final product of that process of accepting an electron onto oxygen will be production of water. As carbon-containing carbohydrate molecules are oxidized, CO2 is given off, so they get smaller and smaller because the carbon is coming from them. So CO2 is produced, as you can see here, at several stages here along the way. And the molecule gets smaller and smaller. In fact, in terms of acetyl-CoA, it is completely used up in this process And I'm going to go into more detail on that in the next few slides. So let's start with the first stage, glycolysis, oxidation of glucose to pyruvate or pyruvic acid, which in terms of the carbon skeleton, the glucose, the carbohydrate, the glucose gets converted into two pyruvate or pyruvic acid molecules. In terms of high energy compound molecules, ATP and NADH are produced. Now there are many stages in this process. All you have to know are the things that I mentioned. You don't have to know all the details necessarily on these slides. And the slide could be, I suppose, intimidating, but what you have to do is have some confidence that I will ask you about and expect you to know about what I emphasize. And that is not all of these names listed here and here and here, or the structures, or where the phosphate is, or whatever. None of that. Okay? And the next slide shows, I'm not going to go to the next slide because it's difficult to go back. But you don't need to know or memorize all that. Just stick with what I talk about. We're talking about glycolysis, and in fact what we're talking about is the first stage or preparatory stage of glycolysis. So glycolysis is the oxidation of glucose to pyruvic acid or pyruvate. Glucose is a 6-carbon molecule. I'm going to write that on the side. Six carbons. There are 6 carbons, and that shouldn't shock anyone. 6 carbons in glucose, and they're shown here as little spheres. 6, that's all. 6 carbons. As it goes along, in the first stage actually uses energy. It doesn't generate energy, it uses up energy. And that's why, and that's shown very clearly here, you can see that ATP is being used up. Okay? See, ATP is being converted into ADP. So in other words, energy is being put in. That's the first stage. But when you get to the second stage, what happens is that ATP gets produced. So we had 2 ATPs being used earlier, but now we have 4 ATPs being produced. That's a surplus net of 2 ATP molecules. Don't worry about all the other details here. In the end, in terms of high energy compounds produced, you have 4 ATPs in the second stage being produced, 2 ATPs being used in the first stage. So 4 minus 2 is equal to 2. So for every glucose that undergoes oxidation to pyruvic acid, you get 2 ATP molecules. And as you can see at the top there, you also get not only 2 ATPs, but you also get 2 NADHs produced. So glycolysis, which is glucose oxidation to pyruvic acid, involves the oxidation of glucose and production of 2 ATPs and 2 NADHs. Did I say NAD? It's NADH. That's an H. The end result of this, in terms of the carbon-containing skeleton, the organic molecule that contains carbon, is 2 pyruvate molecules, 2 pyruvic acid molecules. Pyruvic acid has 3 carbons in it. So pyruvic acid has 3 carbons. We started with one 6-carbon molecule, a glucose, and ended up with 2 3-carbon pyruvic acid molecules. 2 times 3 is 6, right? Okay, we go from a 6-carbon molecule to 2 3-carbon pyruvate or pyruvic acid molecules in the oxidation of glucose during glycolysis, and we have production of 2 ATPs and 2 NADHs. All the rest forget about, don't worry about it. Yes, there are 10 stages, that's all. And each of these stages, yes, there is an enzyme, each of these represents some kind of very specific enzyme that is catalyzing the reaction. It's enough for you to know that there are 10 enzyme catalyzed steps in glycolysis. You don't need to know what the names of the enzymes are. You don't need to know what the names of each of these, intermediate molecules are. Don't worry about it. That's glycolysis. And this is the overall equation for it, but you don't have to know it. It's not in red ink. I wrote it out and it's not in red ink, but you do actually know it because I said you have, a glucose undergoing oxidation, and glucose is 6 carbons to produce 2 pyruvic acids, which are 3 carbon each. That's where the 6 carbons are. Plus you have some ATP produced, right? And you have some NADH produced, right? It says 4. Yeah. Okay, let's move on. There are, in some bacteria, other pathways for the oxidation of glucose. Glycolysis is not the only way that glucose can be broken down and used to generate energy, but we don't have to go into that at all. You don't even have to know these names. Don't worry about them. There are other ways. There are alternatives. So here we are. We've talked about in cellular respiration the first stage or first step in this process called glycolysis, where you get production of ATP and production of NADH, and from one glucose you get 2 pyruvic acids. The next step was pyruvate or pyruvic acid oxidation. So you get oxidation of pyruvic acid, which was produced in glycolysis, and gets oxidized to form acetyl-CoA. Now remember that glucose, originally we started with 1 glucose, 6 carbons. We went to 2 pyruvic acids, 2 3-carbon pyruvic acids, and here we have acetyl-CoA, which has 2 carbons. What happened to the other carbon? Well, it was given off, it was released as carbon dioxide. So whenever there's some, reduction in the number of carbons, total carbons, you can be rest assured it got released as carbon dioxide gas. Gee, what happens to that carbon dioxide? Well, that's what cells release when they are metabolically active. That's what our cells release when they're metabolically active. They release it to the bloodstream, and blood picks it up, carries it to the lungs, and you blow it off. Exactly this same process is happening in your cells in your body, but it can also occur in many bacteria. And along the way, NADH is also produced. So just to sum things up, first stage was glycolysis, oxidation of glucose to 2 pyruvic acids, and production of NADH and ATP. And the next one was pyruvic acid oxidation, conversion of pyruvic acid to acetyl-CoA, and we had going from a 3-carbon to a 2-carbon, We had 2 3-carbon pyruvic acids. We end up with 2 2-carbon acetyl-CoA molecules. The rest of the other carbon is lost as CO2, and NADH is produced. That's pyruvic acid oxidation to form acetyl-CoA. Now, what happens to the acetyl-CoA? We go to the 3rd cycle, and you get oxidation of acetyl-CoA. And it's a full oxidation, and you'll see what I mean. And in terms of energy, you get production of ATP, NADH, and FADH2. Now, this oxidation of acetyl-CoA has a name. It actually goes by several names, and students get confused because they start listening to other sources, looking things up, and they start getting confused because it's being called something else, but looks like it's the same. Well, they're all the same. Krebs cycle, tricarboxylic acid cycle, TCA cycle, citric acid cycle, they're all the same. They all refer to oxidation of acetyl-CoA. And I'm going to stick with Krebs cycle. Wow, this is intimidating. However, you don't have to know all these details. All you need to know is that at the top here, that's where acetyl-CoA enters into the cycle. All you need to do is know what I emphasize, and that is that one molecule of acetyl-CoA, mind you, if you think about the original glucose, which was one 6-carbon molecule, and it went to form 2 3-carbon pyruvate molecules, and then that went to 2 2-carbon acetyl-CoA molecules. Co stands for coenzyme. I won't worry about it, but acetyl-CoA. Acetyl-CoA is a 2-carbon molecule. That 2-carbon molecule is constantly being fed in as a substrate into reactions which have many steps. Each step is catalyzed by a different enzyme. That's what that little blue circle is with a number. Don't worry about them. Know that there's several reactions involved here, but you don't even have to know how many. Just that there's a conversion, each catalyzed by a different enzyme. So acetyl-CoA is converted into citric acid, but it's converted, that's 2-carbon acetyl-CoA is converted because it joins up in that biochemical reaction. It joins up the 2-carbon acetyl-CoA, joins with, or is joined to, this molecule here, which has 4 carbons. Now, 4 plus 2 equals 6. Okay? And that's that molecule there. If I don't say the name, you don't have to know it. All you have to do is know that the 2-carbon acetyl-CoA is being formed into a 6-carbon molecule during the Krebs cycle. So it goes into the Krebs cycle, it joins together with this 4-carbon molecule, so 2 plus 4 is 6, and it undergoes a series of enzyme reactions, enzyme catalyzed reactions, and along the way, and you don't even have to know at which step, okay, but along the way you get production of and release of NADH, FADH2, and some ATP. You end up, as it goes around through all these conversions here, and then you end up with this 4-carbon molecule. Well, we started here, at this point it was 6, and now it's 4. Obviously some carbon was given off. In what form? as carbon dioxide. You don't have to know where, at which exact stage the carbon dioxide is produced. You don't have to know where at exactly which stage the NADH is produced, or the ATP or the FADH. You don't have to know the names of any of these intermediates or any of the enzymes. None of it. All you have to know is that, yeah, some CO2 is produced. What happens to that CO2? Well, if it's a bacteria, it just diffuses out. Maybe it diffuses into your orange juice, which was sitting around on the counter instead of in the fridge, and someone drank from it with their mouth even and contaminated with bacteria. And now it's got some bubbly CO2. Well, it's bubbly with CO2, and you feel it on your tongue when you drink it because it's formed from bacteria that are releasing that CO2. If we're talking about a human, the human cells are carrying out the Krebs cycle, releasing the CO2. It diffuses out of the cell into the interstitial fluid, into the blood, gets carried in the lungs, and you breathe it off. All you need to know is that carbohydrates like glucose can be used by cells like bacteria or mammalian cells in cellular respiration. Glucose is converted by glucose oxidation to pyruvate. That's called glycolysis. Pyruvate undergoes oxidation. That's pyruvate oxidation to acetyl-CoA. Acetyl-CoA goes into the Krebs cycle, and through the Krebs cycle it keeps going round and round and round. And as long as there's more new acetyl-CoA being produced and feeding in, this process is occurring, and NADH is produced, and FADH2 and ATP. Okay, sounds good so far. So we have, as I said, glycolysis, oxidation of glucose to pyruvic acid oxidation to acetyl-CoA, acetyl-CoA in the Krebs cycle. Along the way in glycolysis, we have NADH and ATP. In pyruvate oxidation, we have NADH. In the Krebs cycle, we have ATP, NADH, and FADH2 produced. NADH and FADH produced in 1 and 2 and 3 here, glycolysis, pyruvate oxidation, Krebs cycle. The 3 should be really here, the Krebs cycle. All of that, the NADH and the FADH2 goes to and is used in the electron transport chain along with chemiosmosis to produce a lot of ATP. And I guess, I know, the next slides will be on that process of the electron transport chain. So the electron transport chain, or ETC, involves oxidation of molecules that carry a lot of energy, a lot of electrons. NADH and FADH2, they are high energy molecules, and they can be used for energy production in the electron transport chain, energy in the form of ATP. So the electrons in NADH and FADH2, which were picked up earlier, are now used to produce ATP. And it's used in a interesting kind of, at least I think it is interesting, but it's used in a series of reactions that involve carriers, redox reactions, oxidation reduction reactions, and chemiosmosis. The electron transport chain refers to a series of carrier molecules that pick up an electron and then pass it along. So they're picking it up and releasing it, picking it up, releasing it. Oxidation is a loss, reduction is a gain. So NADH and FADH2 lose electrons. The first molecule in the electron transport chain picks it up, then transfers it over to the next one, and then that one transfers it to the next one, etc., etc., etc. So that's a series of redox reactions. The energy that is released during that process of redox reactions, as those electrons get transferred along, there's some energy released, and it is used by a mechanism called chemiosmosis to produce ATP. And that process of ATP production by chemiosmosis involving the electron transport chain is called oxidative phosphorylation. It occurs in mitochondria. So we're talking about this, all of this. And the next few slides look into that. What you see here is the inner mitochondrial membrane. Remember, I told you mitochondria have an outer membrane and an inner membrane. Okay, so there is an outer membrane and an inner membrane. The outer membrane is out here. Let's draw it like this as an outer membrane. It's a phospholipid bilayer, just like this is the inner membrane. Here is a phospholipid bilayer. Okay, there's a phospholipid bilayer. This is the space between the inner and the outer mitochondrial membranes. Each of these molecules that are embedded, these proteins that are embedded in the inner membrane, are involved in the electron transport chain. They are, in fact, the electron transport chain, and they're involved in the process of chemiosmosis. Okay, let's just label this one here the outer mitochondrial membrane. I'm just going to write outer because it's not easy to write this. Okay, so here we have a series of, don't worry about the name, don't worry how many there are, don't worry about what it's called, don't worry about how it happens. Just know that it happens that NADH transfers an electron to the first one, and that then transfers electrons to the next one. Let's change color here. So the NADH transfers electrons to the first one, and then that one transfers electrons to the next one, and on, and it's a transfer of electrons along the way. So these are oxidation, reduction reactions. Oxidation is a loss, reduction is a gain. Then that one gained, the one that gained loses, etc., 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 until we get to the final electron acceptor, which is oxygen. So oxygen is the final electron acceptor in the electron transport chain. And as it picks up the electrons, you get formation of what's called metabolic water. What about this term, chemiosmosis? What does that refer to? Well, as the electron transport chain is passing along these electrons, what happens is that hydrogen ions are being pumped into the space between the inner and outer mitochondrial membrane. So you get an accumulation of hydrogen ions in this space. Don't worry about how many, don't worry, just hydrogen ions. And this concentration gradient, increase in concentration of hydrogen ions, they move down back through the inner membrane through a protein called ATP synthase. Okay, this molecule ends in A's, so it's an enzyme, and for once they've named it after what it does. It synthesizes, or helps synthesize, it catalyzes the synthesis of ATP. And that's chemiosmosis, the process of chemiosmosis, where you get movement of hydrogen ions in the space between the membranes. Then they go down through the ATP synthase and ATP is formed. Okay? So that was aerobic cellular respiration. It is possible to have anaerobic respiration. It's called aerobic respiration because the final electron acceptor is oxygen, so it's aerobic. It is possible to have respiration that's anaerobic that doesn't involve oxygen. And there are some examples of that, but I'm not going to get into them. And so we had 4 pathways, 4 stages in aerobic respiration or cellular respiration: glycolysis, pyruvate oxidation, pyruvate oxidation, the Krebs cycle, and the electron transport chain and chemiosmosis. And where do they occur? Well, it depends if we're looking at a eukaryotic cell or a prokaryotic cell. Eukaryotic cells have organelles, membrane-bound organelles. There's a nucleus, there's a mitochondria, there's endoplasmic reticulum, there's Golgi apparatus, etc., etc. Prokaryotic cells do not have any internal membrane-bound organelles. Okay, let's review. You knew that. So if I said earlier that it had the Krebs cycle is, that the electron transport chain is in the inner membrane of mitochondria, I was talking about eukaryotic cells. If it was a bacteria, it would be right at the single membrane, the surface cell membrane. So these things here in a prokaryotic organism, these 3 occur in the cytoplasm, and the electron transport chain is present or is occurring in the plasma membrane. In eukaryotes, the first 2 are in the cytoplasm. The Krebs cycle is occurring inside the mitochondria, and the electron transport chain is in the inner membrane. Okay? And here you see a diagram of mitochondria. Obviously, it's taken from a eukaryotic cell. It's a subpart, a membrane-bound organelle. Remember that the mitochondria have 2 membranes, an inner membrane and an outer membrane, just like gram-negative organisms have 2 cell membranes. There's the inner and outer membrane. The matrix is the space internal to inside of the inner membrane. Aerobic respiration generates quite a bit of high energy carrier molecules. You get, with cycling through it, you get complete oxidation of a molecule of glucose. It is an aerobic respiration process. You get ATP produced, and you get NADH and FADH2 produced. This you don't have to learn. It's blue, I realize, but let's look at it as being in black ink. You don't have to memorize this. You already knew that these 3 high energy molecules are produced, and you know that 2 of them, the NADH and FADH2, will be used in the electron transport chain to generate ATP. Okay, so actually there's quite a bit of NADH, and there's some FADH2, which is produced in the Krebs cycle, produced. It gets used in the following way. Remember from the previous, again, I know it's blue, but I meant it to be black. Here you have, remember we had 10 NADHs produced, and you end up with 30 ATPs, and that is because 1 NADH can generate 3 ATPs. 3 ATPs will be generated in the electron transport chain per NADH, and 2 ATPs will be produced from per FADH2 produced, and that's why you have 4. Suffice it to say, the end result is that you get production from the full breakdown oxidation of 1,6-carbon glucose. You get in the end 38 ATPs. That's in a prokaryotic cell, bacteria. It's less in eukaryotic cells because a lot of the ATP is produced in the mitochondria, right? And it takes some energy to move the electrons that are involved across the mitochondrial membrane. To move high energy compounds across the mitochondrial membrane takes some energy, so 36 ATPs are produced per glucose molecule metabolized. Per oxidation of 1 glucose, you get 38 ATPs in prokaryotes and 36 in eukaryotes, because in eukaryotes you have to move across the stuff across the mitochondrial membrane. Okay, I think that's enough. This is the end of part 1, and after this that's all of cellular respiration, oxidative aerobic respiration, and oxidative phosphorylation that's occurring due to chemiosmosis and electron transport chain. And for the next part, we'll talk about fermentation.